All right, great. So welcome everyone. It's my great pleasure to introduce Dr. Samantha Santacruz, who's visiting us from the University of Texas at Austin, where she is an assistant professor in the Department of Biomedical Engineering. She's also affiliated with neuroscience as well as electrical and computer engineering there as well. And so she's a really great example of someone that bridges engineering and neuroscience work. And she's going to tell us today about some of her really exciting research on neural interface technologies for both basic science applications and for therapies. Right.
Thank you. Thank you, Amy. It's my pleasure to be visiting University of Washington for the very first time. It's also very nice to take a break from the heat in Texas. If you look now, it's in the cool 80s, but last week we were already in the 90s, so this is pretty refreshing. But I'm very happy to be here and talk to you guys about my work in neuroengineering. So as Amy mentioned, I work on developing brain-machine interfaces for a variety of different applications, and really kind of engineering the advancements of neuroprosthetics for a wide range of neurological disorders. So that's what I hope to talk to you guys about today. So just a little bit about me, because I think I'm new to a lot of you. So I'm from California, just a few states down from here, from a tiny little beach town. And I've just been bouncing back and forth between California and Texas. So I did my undergrad at Berkeley in applied math, and then I went to Rice for double-E. Back to Berkeley for my postdoc. That's a connection that Amy and I have. I did my postdoc in the lab where she did her PhD. And kind of in my PhD and postdoc timeframe is when I really started focusing on engineering problems within neuroscience and working in this area. And then now I started my own lab at UT Austin, primarily based in biomedical engineering, where we focus on developing these brain-machine interfaces for a range of applications. And in particular, we're really focusing on accessing the brain at different scales so that we can understand what's going on across scales within the brain as it relates to behavior and to diseases. So we set out on this path of using experimental and technological approaches in animal models to study causal and correlational relationships between neural activity and behavior. The goal of this is to help us ultimately determine some biomarkers that are disease-related that could help us develop therapies, say like closed-loop stimulation paradigms, but also just to understand the neural correlates of behavior generally. So we know that there's this scale and organization across the brain, across multiple different levels. And so we're really interested in understanding what's happening with individual cells, how they're organized into neural circuits, and how things are being shared, information is being shared across different brain structures, and then how that ultimately relates to a functional output like observable behavior that we're seeing. So one of the big goals in our lab is to develop neurotechnologies to record activity at multiple scales, and that's really what we're going to focus on talking about today. And then another one of our goals is to implement these neurotechnologies in novel ways in order to develop BMI, or brain-machine interface paradigms, like closed-loop stimulation or just closed-loop control. So one of the big challenges in the neural technologies that we have is that they're limited in terms of the functionality that they have. And the two main functions that we're interested in are reading and writing. That's the way that I like to think about it. So for reading, what we mean is recording some neural activity and being able to read out that signal. And so we often think about that in the context of a neurotherapy in terms of motor neuroprosthetics, or what a lot of people generally refer to as brain-computer interfaces or brain-machine interfaces. So the idea with motor neuroprosthetics is that you implant some electrodes. Oftentimes we're using invasive technologies, but you can also do this with non-invasive methods as well, like EEG. But you're recording some neural signal. If you have an implanted electrode, then you're generally looking at a broadband signal and perhaps focusing on either the low frequency content of that, so looking at the local field potential, or the high frequency activity where you're seeing the action potentials from individual neurons. But the idea is that you would take some signal and you would map that to a control signal. So you're reading out the neural activity. In the bottom left diagram, we're reading out spikes. We're mapping that, or we're taking that into our decoder, which is just a mathematical mapping. And the output of that is going to be a control signal that's updating, say, like the joints of a robotic arm or velocity of a cursor on a screen, something like that. So far here, I would say this is really like a reading out technology. We think of this implemented BMI as closed loop because there is visual feedback in the system that's kind of closing that loop. But in terms of the technology that we need, we only need something to be able to record out those signals. But another function that we might be interested in is writing in new information. And the main way in which we think about doing this is through stimulation methods. So electrical stimulation, I think, is still one of the most common ways to do this, especially because it's a way that translates to humans easily. But you can also do this with magnetic stimulation or optical stimulation and other methods as well. And the idea is that you're mediating the activity in the brain in some way through this intervention. And so we think of that as like writing in new information because you're perturbing the activity in a way that's changing representations in the brain. So we're really interested in neurotechnologies that can achieve both of these things, ideally within the same device, but this doesn't always happen. But so we're interested in materials and device design that can achieve these two general goals. So one of the challenges to doing this and achieving this goal is that conventional materials that we typically use for neural interfaces are limited in terms of what they can achieve. So we often use metal materials, for example, for our recording materials. And these metals tend to be very rigid. For those people that have touched brain and interact with brain regularly, you know the brain is very soft. So you could think of it like Jell-O. Not as delicious as that, but some people like eating brain. But so there's a mismatch there in terms of the mechanical properties, right? If we've got these rigid metal materials that we're putting into a really soft material, we can potentially cause more damage to that neural tissue because of that mechanical mismatch. So think about sticking a needle into Jell-O and like wiggling it around. So that's one challenge that we have with these standard materials. The other is that not all metals are really good at charge transfer, so injecting current into the brain. So they have poor electrochemical properties that prohibit them from being as appropriate for doing electrical stimulation. And then the other thing is that if you're trying to get both of these into one technology, there tends to be in these standard materials this inverse relationship between contact size and impedance. So the smaller contact you have in your electrode, the finer spatial resolution you're getting, and the easier it is to resolve activity from, say, like an individual neuron. But at the same time, the smaller that you're making it, you're getting a higher and higher impedance, which makes it a worse material or worse electrode for doing stimulation. So it's harder to pass current through if you have a high impedance. So often we have this trade-off between picking the right materials to do stimulation and picking the right materials to get good neural recording. So one of our attempts to address these is to work with carbon nanotube fibers. So carbon nanotubes fibers are slightly different from carbon fibers in that we fabricate them through a wet spinning process using carbon nanotubes. So we have carbon nanotubes that are in this superacid solution shown on the very left. And through this wet spinning process, we extrude them into a coagulant bath through a small pinhole. And we just put tension on that line of carbon nanotubes as they're coming through to create this thread-like fiber. The way that we control the diameter is basically by the size of the pinhole and how much tension you're putting on that line. But so it means that you can fabricate fibers in a range of diameters using this technique, and it really depends on kind of what you're targeting, what size fiber you might want to make. But so you end up with this spool of carbon nanotube fibers that are really nice because they have great electrochemical properties, but they're also really strong while being very flexible. So this makes them a potentially ideal material to use for both recording and stimulation. So now to make an electrode, all you really need to do is to insulate along the length of this, just have the end exposed, and connect the other end to your electro-recording system. So we started out doing this, making electrodes that had a small number of channels. So here's my little thing. So here we're showing a stereotrode, which is a 2-channel electrode. So stereotrodes are commonly used for doing stimulation. If you want to do bipolar stimulation, you're stimulating between 2 electrical contacts. So we made our stereotrode by insulating 2 fibers, just twisting them together, heating the insulation up a little bit so that they would kind of stick together, and then exposing the tips. And then we also made some tetrodes as well, which are 4-channel electrodes that people commonly use when they're recording from areas like the hippocampus or where you have like really high density of cells. So this is work that was done with Flavia Vitali, who's now at UPenn, and people who are all at Rice, Matteo Pasquale, Calo Camiri, and Behnam Ajeng. So we said that carbon nanotube fibers might have these ideal properties for being able to do both read and write out functionalities. So the first thing that we're looking at is just whether or not we can break that relationship between contact size and impedance. And so what we found is that with the carbon nanotube fibers, if you kind of cut it to make your exposed area and look at that cross section, it's not smooth. It's really rough because the fiber itself is made up of all of those carbon nanotubes. And so that means, well, the cross-section area is really small. The effective surface area is actually quite large because the surface is not smooth. So we can have a surface area that's much smaller than what we would say have in a standard platinum iridium electrode, which is a common material used for stimulation. But we have a much lower specific impedance. So we no longer have that same relationship. We compared the charge injection capacity of our material with some other common materials that are used for stimulation. So charge injection capacity is a metric that we used in order to characterize how well we can pass charge through this material into our tissue. And our carbon nanotube fibers are the second most darkest blue here. So we see that it does really well compared to some standard materials, and in particular materials that we see commonly used in humans for deep brain stimulation technologies. The other thing that we said was that these standard metals are really rigid and there's this mechanical mismatch, so there's likely that we're doing more damage. And so we can try and characterize that by looking at the immunoresponse of our electrodes compared to other standard materials to see if there's a lower immunoresponse, which would indicate potentially that we're irritating the brain less and doing less damage. So we did a study where we implanted our electrodes with some controls that, again, were just kind of like off-the-shelf standard electrodes that people would use in their neuroscience experiments. We had them implant them in rats for 6 weeks. And then we looked at postmortem histology, looking for markers like astrocytes, microglia, activated macrophages. Here we're showing activated macrophages, which are a marker that relate to having a high inflammatory response. And we see that there's a much lower presentation of activated macrophages for the carbon nanotube fibers compared to the platinum iridium. And certainly part of that can be attributed to just the difference in size. It's hard to decouple those two because we can't size match the platinum iridium to our fibers. But overall, we do see a lower inflammatory response with our carbon nanotube fibers compared to this other standard material. So it looks pretty good for doing stimulation here. The electrode is going to have a low impedance, it's going to have high charge injection capacity, and it's got a low inflammatory response, which means we're potentially going to be able to keep it in the tissue for a long time and keep the tissue happy. We also looked at the recording capability. So here I'm showing an example of one of the tetrodes that we made. So here one channel is the carbon nanotube fiber, and the other channels are nichrome wire that are electroplated with gold. So again, this is kind of like a standard way that people might fabricate tetrodes for, say, recording from the hippocampus. So we just replaced one channel with the carbon nanotube fiber so that we could record across all these channels and essentially record from them that are co-located and compare the neural recordings across our channels. So we saw that the integrity of our neural recordings is really comparable between the carbon nanotube fiber and the nichrome wire recordings that we were seeing. So in theory, the stimulation properties are good. The recording properties are also good. But we wanted to test out if this holds true in actual behavioral experiments. So we tested these out in hemi-Parkinsonian rodents. So hemi-Parkinsonian rodents are rodents that have been lesioned in one hemisphere to mimic Parkinson's disease just in that hemisphere. So here we have a stain for tyrosine hydroxylase, which is a protein that is specific to dopaminergic cells. So you can see that it's just appearing in one hemisphere and the other hemisphere is lesioned. So for our animals that we use in this study, on average, they had about 86% cell loss. And so what you can do with these animals is you have a built-in control for basically like healthy motor behavior and Parkinsonian motor behavior. And deep brain stimulation is a common therapy that is used to treat Parkinson's symptoms. So we can implant our stereotrodes and do bipolar stimulation and see how well this is treating the Parkinson's symptoms compared to platinum iridium. And so one of the behavioral assays that we did is this, rotation test. So if you put the hemi-Parkinsonian rodent into this cylindrical environment, the natural tendency is to rotate in one direction. And this is because they're lesioned on one side. So you see this like rotational behavior where he's always going to the right. And then we turn on stimulation. And you can see initially he's not rotating as much. This is a weaker level of stimulation, but we still do see when he's moving that he tends to still move on the right side. So similar to like pharmacological therapies, stimulation is something you need to titrate. You need to figure out the best parameters for treatment. Here we turned on higher frequency stimulation, and initially it's inducing rotations in the opposite direction, but then we see as he adjusts that he stops rotating altogether. So this is one of the behavioral assays that we did looking at the efficacy of deep brain stimulation in these hemi-Parkinsonian models. And we compared the efficacy of our carbon nanotube fiber electrodes with platinum iridium, which is our main control. And so the way that we characterize how well we're stimulating is by how much we've reduced the rotations. Because if you didn't have Parkinson's, if those rats didn't have Parkinson's at all, they wouldn't be rotating. So we see that when DBS is off, we've got a comparable number of rotations as they should be, and the amount that it's reduced is about the same between our two recording technologies. So what this means is that the carbon nanotube fiber isn't really outperforming our controls, like our nichrome wire for recording and then our platinum iridium for stimulation, but it's doing something that neither of them can do in one thing, right? It's able to do both functionalities just as well, whereas if you were trying to do stimulation with nichrome, it wouldn't work as well, and if you were trying to do recording with platinum iridium, it also probably wouldn't look quite as nice. So the examples that I showed with the carbon nanotube fiber, we focused on just doing those small channel counts. So we were doing 2-channel or 4-channel electrodes. And that's because our main fabrication method was just take a bunch of fibers and spin them together, which is how people will commonly do like tetrode fabrication. But it's not something that really scales up. And if you've been paying attention in the neurotechnology space, people are really excited now about potentially thousands of channels. We're all about like getting the data. So we really wanted to focus on microfabrication and microassembly techniques that would allow us to scale up the channel count and do things that could be more high throughput. But like anything, you have to start somewhere. So I'm going to show you some work where we worked with carbon fiber arrays, starting with 32 channel count, but really focusing on developing those microfabrication and microassembly processes so that they could ultimately scale up to larger and larger channel counts. So this is work that was done with Travis Massey, who's now at Lawrence Livermore National Labs, with Jose Carmena and Michelle Maharbitz, who are at IOTA Biosciences, and Chris Pister, who's at UC Berkeley. So we developed these methods of microfabrication. That's kind of summarized over here, but a range of etching and deposition and all the fun stuff to create our final substrate in which we can load in our carbon fibers and create our array. The carbon fiber loading is the main part for us that is still manually done, so we have to assemble those ourselves. But otherwise, everything is more automated. To add in the fibers to the substrate that we've microfabricated, we load in the fibers through an alignment substrate. So this is what like the end of our array looks like. It's 6 by 6 with those corners not being active channels. So it's a 32 channel array. And what we do is we create kind of like a matching alignment substrate that has holes in exactly the same position with the same pitch. And we put them on top of each other, and then we load in our fibers, and then we raise up our alignment substrate here to ensure that our fibers are parallel to each other. So essentially, it's like we're combing through those fibers to make sure that they're staying apart the same distance all the way to the length that we want. And then we fix our fibers in place using a silver epoxy and cure that, and then trim our carbon fibers to the length that we want. And we actually do that using a microtome, so that we're cutting them all to the same length. We plated this with PDOT, and we can see that nicely reduces our impedance to make this nice for recording. And then we tested our carbon fiber arrays also in rodents. So we implanted these. I should note that these are like super high density electrodes, so the pitch is very small between them. So similar to something like the neuropixel that you guys may have heard of, you tend to see the same neuron across multiple channels because of how close together those channels are. So we recorded from our neurons. We can see that we get nicely looking action potentials. And what we saw was that there's the units that we're recording is basically like just one or a few other units adding in. So they're all super synchronized because we're getting the same unit across everywhere. And we did, let's see, you can't really see it there. Yeah, we did basically like a heat map study to show like you could locate where that unit was and see like the peak to trough ratio dropping off from that location from the other recording sites. So we did a lot of work to make sure that this wasn't artifactual. It was just the same unit being recorded across all of our sites. But so we showed that we can use these microfabrication and microassembly techniques to build up this style of arrays, and you incorporate our newer non-metal technologies for recording. So this is the highest density microwire style electrode array to date, and this technique is scalable to a larger number of electrodes. So this was 32 channel. So this is kind of one of the things that we're working on now is growing this to higher channel counts. I don't know how many of you here work in microfabrication, but as you know, one clean room is not equal to another clean room. So we've had to change some of the things in our microfabrication processes just because the clean room at UT has different tools than the clean room at Berkeley and different materials. So we spent a lot of time kind of re-optimizing some of those processes to scale this up. We're now at the point where we're kind of happy with our substrate, and we're about to start loading in our fibers. We still need to figure out how we're going to insulate things because that needs to be changed as well. But the ultimate goal is to fabricate this, this array that is going to have 64 channels, but it's also going to be longer. So one of the things that's also missing from kind of the neurotechnologies that we have available to us are technologies that can go chronically to deep brain areas. So a lot of the innovation in neurotechnology is focused on cortical interfaces, which has been really fantastic. But it means that we're ignoring some of the deep brain structures that are still really challenging to get to if you want to do chronic studies in those areas. So we're focusing on those, and we'll be testing those out in non-human primates, which is the main model system that we're working in right now. And I just want to highlight that Megan Baker is this amazing student that I recruited from Purdue who's leading these projects in the lab currently. Another technology that is like an ongoing project that we're working on right now is developing these really thin platinum ditelluride probes. We're starting out doing a cortical version, so recording from superficial cortex, and then these will be scaled up for deeper recordings. The platinum ditelluride is nice because it grows easily on these flexible substrates, and it's super thin, so while these things are wide, the width is very tiny. And this is something that we're working on with our collaborator, Deji Akiwande, who's at UT Austin. And in the process of doing this, we've gotten kind of like more and more focused on really trying to optimize our implantation strategies. So these materials that we're working with, we're always trying to keep them more flexible or softer because we want them to match the mechanical properties of the brain so that we're doing less damage and we can have them chronically implanted and maintain good signal over time. But what are the mechanical properties of the brain? So we had been using agarose gel as a model and then a composite hydrogel, and we found that there's really kind of limited data out there on non-human primate neural tissue, and especially how mechanical properties of the brain might differ across structures. So like how cortex differs from white matter and deep brain structures, which are all things we have to traverse with our chronic implants. So we've been working with our collaborator, Manuel Rausch, to do some tissue mechanics experiments on explanted, unfixed NHP tissue so that we can optimize our composite hydrogel model and create a really good mimic for doing test implantations with our different electrodes and optimize our implantation strategies for these electrodes. I would say if you don't work in this space, I would say making the electrode is like a PhD on its own. Figuring out how to get it in the brain is almost another PhD on its own because it's so hard. And thankfully, we have a student who's willing to take on that challenge. So this is Grace Jean-Pierre. She's leading these efforts with our collaborators. Something else that I wanted to tell you that's also ongoing related to our neurotechnology efforts is doing some electrochemistry in the brain. So I focused a lot on like electrophysiology and electrostimulation, but we're really interested in being able to also characterize what's going on at like the chemical scale in the brain. And in particular, we're interested in what's going on with dopamine. And this is because we're focused a lot on behaviors where dopamine impacts behavior. So we're fabricating carbon fibers for recording from the non-human primates and recording the dopamine signal. The carbon fiber design is not really innovative. We're learning from the masters and how to develop these and just kind of optimizing the specs based on where we're targeting in our NHPs. But the idea is that we have a carbon fiber that's just at the tip. That's where we're doing our recording. And then we have this like outer, what we call like a guide tube that we're lowering the electrode through that helps us like penetrate the brain. But for us, our guide tube is also electrically conductive, and we're using that as our reference electrode. So we have it really controlled the distance between our carbon fiber and our reference electrode when we're doing these recordings. We're doing the recordings of dopamine in the striatum. So we've done some testing to show you get signal in response to things that are repetitive, like getting juice. You can also stimulate and get dopamine release in striatum. And our ultimate goal is to see if we can use some neurofeedback paradigms to modulate dopamine release and being able to measure that using our cyclic voltammetry techniques. So we developed a closed loop brain-machine interface paradigm where we're recording from monkey, where we're focusing on recording local field potential signals and looking at power in specific frequency bands, so especially in delta and beta band, and looking at a function of those things, and then having the animal move a cursor to specific targets, where each of these targets that are shown, this square target on the screen, is based on a specific level of delta or beta power. So basically, like to hit this target, he has to modulate beta really, really high, like we're seeing in this red trace here. So around 0 is the target hit. So leading up to moving the cursor there, you can see he's ramping up the beta power and then hits the target and then goes down. And we have 4 different levels in which the animal has to do this. And so the goal is to see if by modulating some neural correlates of dopamine release, if we can actually see a change in dopamine release. So this is something that's ongoing that we're looking at. And this is work that's being done in collaboration with some clinicians, clinical collaborators, because we're going to do this as well in humans in parallel. And then with our colleagues from Wake Forest and University of Zurich. So in parallel with our monkey experiments, we're also going to have humans do an fMRI neurofeedback experiment where they modulate bold signal in SNVTA and try to do the same thing, like up or down modulate. And then we'll recruit DBS patients, deep brain stimulation patients, to do this task preoperatively. When they're in the OR getting their DBS electrode implanted, we'll have them do the cyclic voltammetry and we'll measure dopamine in humans. And this is where Ken really comes in because he's a superstar in doing these type of measurements in humans. So this is a project that is very big and is being led by our student Lou, along with Hannah and E, who are doing this. So I focus a lot on the technologies for looking at the electrical activity, and then right now for doing electrochemical sensing. But we're also interested in what's happening across different scales. So we've kind of covered like spikes and local field potentials and then chemicals. But one of the nice things with doing imaging is that you can see activity from individual units, but you can also see how those units relate to each other, which we never really know when we're doing electrophysiology, right? So even when we have electrodes and we know where those electrodes are placed, if we're recording from one unit here and one unit here, we have no idea like what are the actual relationships between those units. And that's one of the things that imaging helps us overcome. So we did oops, it's not playing. Okay. Well, just pretend green is flashing in that black. So we're really interested in trying to characterize the dynamics happening at this scale by using optical imaging technologies, and in particular focusing on one photon imaging. So we chose one photon because there are some limitations associated with two photon imaging. Two photon imaging has had a lot of success, and it's been like the thing that translated easier initially to monkeys. From smaller animal models. In primates, it's really hard to get some of these technologies working just because we work with so few animals generally, right? So it's harder to do those iterations, although maybe you have that problem less here with the primate center. But so we worked on optimizing the one photon calcium imaging setup in the non-human primate to mitigate some of the limitations that we saw with two photon imaging. And in particular, the nice thing that we have is we have this implanted grin lens. So we're imaging at the face of that lens, and that is chronically implanted. So whenever we want to do an imaging session, we just have to mate our mini 1-photon microscope with that implanted thing. But it means that where we're imaging is always fixed. So we never have to do like new alignments or deal with any sort of realignment. Because of this setup, we also don't need to do head restraint. So this is tethered, but the animal is able to freely move their head around while sitting in their primate chair. This is work that we did in collaboration with people at Inscopix, Anil and Jojo, and then Karen Moxon, who's at UC Davis, and you already saw Jose's lovely face. He's at in IOTA now. So in the non-human primates, we did imaging across a period of about 8 months. This was primarily based at the UC Davis Primate Center. We started out doing our injections, doing some preliminary imaging just to make sure that things were going well with our virus injections. Then we trained the animal in our behavioral task, and then we started imaging. In this block from 0 to 76 days, I think we did like 40-odd sessions in total, and 66 sessions in total, and 42 of those were like consecutive days back to back. We were really interested in capturing how stable the recording was across days. So we looked at that, and what we found was that if we looked across days, the number of cells that we were getting every day is pretty stable. And then the signal that we're getting is also stable across days. And then for the calcium signal that we were getting, we looked at these like calcium events, and that also seems to be like pretty consistent across those first 76 days where we were really focusing on. And so in order to characterize like how many neurons we were really maintaining across those sessions, not just saying, okay, it's the same number of neurons, but saying it is the same neurons, we did some stuff with the imaging where we looked at different days and looked at the same, how much overlap there was between the ROIs, so the regions of interest that were identified as being individual cells, in order to track how many cells were being maintained across imaging sessions. So here, for example, is day 29 in purple, and then in green is day 36, so that's 1 week apart. And then if we do an overlay, I think the white ones are all the ones that are the same ROIs between those days, so it's like a 63% overlap between those days. And so we did this for basically like every iteration of like difference of days and how many days within those first like 76 recording sessions. So we found that like across, let's see, for an intersession interval of almost the whole time period, so more than 2 months, we were getting like 40% of cells still maintained from the very beginning. So that was really exciting to us that we were able to maintain cells for such an extended period of time and see that maintenance. Yeah, what's my other stat? Yeah, so we also saw that with a minimum intersession interval of 1 to 4 days, that we had approximately 70% of the same cell pairs maintained. So we did this during behavior. So we trained the animal in this basic reaching task. Okay. So the animal is reaching to one of 2 zones, which we cleverly labeled zone 1 and zone 2. I think in this video he's reaching with just one hand, but we had different flavors of this task where like if he's reaching to the right, he has to use his right. If he's reaching to the left, he has to use his left. Or we had it either way, it's always just using the right arm, et cetera. We did different versions. The implants here, we're showing just data from one hemisphere, but he was implanted in both hemispheres. So the first thing we did was just look to see what kind of like specificity we saw in representation of reach to either direction with these neurons. So we know from EFIS data recording from motor areas that you see this like specificity for moving in particular directions, right? And so we certainly saw that same effect where we would see that some neurons are selective to movement to zone 1, so they really care about moving to that direction. Some were just modulated for reaching, so like when the animal was moving, there was activity, but it didn't matter whether it was to zone 1 or zone 2. And then some are selective to just zone 2, so only for that side is that neuron moving. So we characterized the selectivity of the neurons, and then we looked at how stable that was for the neurons that were maintained over time. And this is something that I think is also harder for us to do with EFAs because we don't really have, beyond like statistical methods, we don't have as much confidence that we're really maintaining exactly the same cell over time, even with a chronic implant. So we looked at the tuning across days to see how well that was maintained, and we also see that it's pretty kind of flat, these curves, which means that the neurons that were selective for particular reach directions tended to stay being selective to that reach direction. So one of the maybe disappointing, but maybe not surprising things that we saw that we looked at was we wanted to look at whether there was like spatial organization of the neurons relative to what they cared about. So for example, you could imagine that the neurons that are more selective to reaching to zone 1 might be like co-located, and neurons that care about moving to zone 2 may be co-located. And that's a question that we would only be able to answer with imaging. We couldn't really answer as well with EFAs. And what we found was it was really like salt and pepper, like they were all mixed together, which is maybe, it was disappointing, but maybe not surprising, right? That like that's how the brain decided to organize and optimize. But that was something that we also looked into. So on this same front of using like genetically encoded sensors, right now we're in the process of optimizing 2 different sensors. One is D-Light, which is a sensor that already exists, like you can buy it. So this is something that has been really successful in rodents, especially in mice, but hasn't been tested out in primates yet. D-Light is a genetically encoded sensor for dopamine, so we could record dopamine using like fiber photometry methods. So we're in the process of doing this, and that's in collaboration with Tommaso Patriarchi and Leif Fenno, who's at Delmed. And then we're also interested in developing some neuropeptide sensors, and those are not on the market right now. These are currently under development in Tommaso's lab, but we're really interested in these things that that also are related to dopamine release and have like mediate dopamine release. So we're in the process of optimizing these in the non-human primate as well, so that we can continue to grow the tool set of things that we have to work with the non-human primates. And these are efforts that are being led by these 2 amazing grad students. So Lou, you saw his smiling face earlier, and then we also have Reina Taniguchi, who's our newest lab member and is co-advised with Leif Fenno. So we've reached the end, and I just want to acknowledge all of my amazing collaborators who have contributed to the results that we packed in here. None of it is possible without all of those really rich and rewarding collaborations across multiple institutions. And then also my lab, who are go along with all the crazy lab activities that I throw at them. And then also the funding that we've gotten. Most recently, I got the Career Award, which was really exciting from NSF. And then I have to end with Hookem because that's the UT thing. These are all the people in the lab, some people you didn't get to see, and then also my greatest output since I've been faculty there as well. But thank you so much for your attention.
What questions do folks have? Yeah, check.
Great talk, Samantha, first of all. I want to try to link together the imaging calcium imaging studies with some of your early electrode reporting. You saw really impressive stability of those cells over time with the imaging. The fact that we see cells changing or disappearing when we're trying to record them on electrodes, do you think that's all about tissue response and stiff bad electrodes, or do you think, like based on what you saw in the imaging, do you think sometimes cells are on one day and gone the next, and then they come back later?
Yeah, I think they're moving around. Yeah, they're not staying still. I think that's part of it. Like so the way that we did our like overlays, like we always did some kind of like motion correction, but then also some correction for like just of the field. But a lot of times, like it would just be like some neurons just migrating slightly over time. And so I think that for electrophysiology, a lot of the kind of loss of cells we see is certainly like related to the immunoresponse and glial response and everything, but I think loss could also just mean things are moving.
Yeah, Steve.
Can you comment a little bit about the advantages of the carbon nanotube versus carbon fiber? Those are different things precisely. So you use both.
Yeah, we use both. So the carbon fiber we chose for the second project because they're just more readily available. So you can just buy carbon fiber, it's really easy, whereas the carbon nanotube fiber needs to be made, or you can get it from Mateo. But yeah, the carbon nanotube fiber, I think ultimately is better for doing both things, for doing stimulation and recording, because you do get that like much lower impedance for the same cross-sectional area. So it tends to have a better like charge injection capacity, but still spatial specificity for recording. But the carbon fiber is also really good, especially if you're like electroplating and using other things to make it nice. So right now with the arrays that we're fabricating with the higher channel counts, we're doing the carbon fiber initially just to kind of like test things out, but we'll probably want to swap back to the carbon nanotube fibers. Yeah.
So I have a question. So I think seeing the array of different, sensors and measurements that you're making is super exciting. And you sort of started with that motivation of, multiscale, right?
Yeah.
So how do you think about the role that each of those different scales of measurements will ultimately play in, a therapy or something like that? How do you combine all those different scales together?
Yeah, I think they each kind of have their own purpose. So for example, in with the voltammetry stuff that we were doing, we are doing multiscale because we're always doing some recording modality in addition to the voltammetry. But ultimately, we're not going to be like chronically measuring dopamine in a human, probably. But I think if we can establish that you can come up with a neurofeedback paradigm that allows you to self-regulate dopamine, then that could become like a really powerful tool where we're not necessarily monitoring dopamine chronically, but maybe establishing it in like, the DBS electrode implantation, and then using that as like an add-on to make DBS more effective or just to make meds more effective. And they learn how to like self-regulate through strategies they found to be successful while doing the fMRI training. So I think like the different scales, it really depends on what's the ultimate goal. If it's a therapy, then you have to think about what's the final output, like what do you want to be impacting and what's clinically like translatable in a feasible way. I think for the neuroscience, that's a much harder question, right? Because like I think it's endless what we could look at across scales and try and relate things to each other, and that to me has been the hardest problem of really like narrowing the field of questions that we're looking at. But yeah, I think we're really interested in seeing if we can establish some like causal relationships between things across scales with like the dopamine and the local field potential stuff. What are some of the other examples that I gave? We are interested in like with the new sensors, like with the D-Light sensor for dopamine, where we'd be interested in like seeing how we can relate that to spiking activity from like individual neurons that are in that area. Because I think also like the kind of knowledge that we have on how like dopamine release relates to like dopaminergic spiking is still also, there's a lot of things to figure out. So combining these things could be really exciting. But across all the scales, that's hard. So hard.
Great. Yeah, Jen.
Just follow up to part of your answer to Amy's question, where you were talking about that intriguing idea of having patients learn to self-regulate their dopamine release. Does your BCI experiment you described like start to get at that? I know you just said that the activity and the release and how it's correlated. Could you with D-light sensors or something, could you actually do biofeedback on real-time dopamine release, maybe in the animal models, and see if we can learn to...
Right, so right now we're trying that with the LFP. So basically like the voltammetry with the LFP neurofeedback, we haven't done that together yet. We've just like debugged those things separately and established that it works. But the next step is to bring those together to see if like modulating the LFP activity does change the dopamine release. And then we have like some tasks where we have aversive and appetitive stimuli that we want to see, like, okay, if the animal trains and like down regulates dopamine, so now like the tonic levels of dopamine are lower, does that affect phasic dopamine release in response to stimuli? So we're going through all of that characterization as well. But the idea with the D-light, the genetically encoded sensor, is that that could be potentially a more stable replacement for the voltammetry, at least in the animal model. But yeah, there was a really cool paper, I don't think it's published yet, but it's on bioRxiv, where they looked at doing D-light and voltammetry in small animal models and relating those signals. And so we're really excited to leverage that for our own stuff as well.
All right, well, let's thank Sam again for a great talk.